Hi class, and welcome to your next video on mutations. So we've talked a lot about mutations in class. We've talked about how there can be changes in the DNA, and when the DNA changes, that is going to change the amino acid that is coded for. And so we want to talk specifically about what the different types of mutations are. So mutations happen when a cell makes a mistake in the DNA. This happens generally during replication, but it can happen at any time when the DNA is being copied. The cells try to avoid making any mistakes in their DNA, but this doesn't always happen. They do have some things that are kind of like a proofreader, but mistakes, just like when you turn in an English paper, are not always caught. So these mistakes are known as mutations. A mutation is any change within the DNA sequence. And the reason this is so bad is because think of it as, remember how we said it's like the master copy? And if you're going to make a copy of the DNA with the mRNA, that copy is still going to have the mutation in it. And then the mRNA is going to go out and make the amino acid sequence from that copy of the DNA which had the mutation in it. So we keep carrying that mutation forward instead of there's no way for us to get rid of it. So the first type is a point mutation, and the point mutation has several types of mutations. They occur at one point in the DNA strand, and the first type of that is a substitution. Substitution basically means we are substituting one nucleic acid base for another nucleic acid base. So think of the sentence, a fat cat sat on a hat. If we change one of the letters, just one, we change the entire meaning of the entire sentence, and we change how we actually think about that sentence. So if we change the letter C on the fat, or the F to a C on the fat, now we have a cat sat on a hat. Doesn't really make sense. It seems like someone duplicated a word here, right? So a point mutation can just be a substitution where we're changing one thing. There's several types of substitution mutations. So the first one is a nonsense. Nonsense means we're changing that one base, and that one base now changes it to actually a stop codon. So remember we said when we get to a stop codon, everything is going to fall off. We're going to stop reading the mRNA, and the protein is going to stop being made. So what happens in a substitution mutation, the protein is prematurely stopped, and we don't get the full structure of the protein that we need. The next type is called a missense. A missense mutation codes for a different amino acid than what originally was coded for. So if we were originally coding for phenylalanine, now we might be coding for methionine. So we're changing that amino acid that we're coding for. And what that does is it actually changes the function of the protein. It can make that protein non-functional. It can make it into a totally different protein. And this is where we start to get some different kinds of disorders due to the proteins that we have or don't have. The last type of a substitution mutation is a silent mutation. And a silent mutation is still where you are changing one of those bases, but nothing is happening with the amino acid. The reason being is, remember how we have 64 codons, but we only have 20 amino acids. So remember that multiple codons are coding for the same amino acid. Well, when we change one of those codons, but they're very similar, they may still code for the exact same amino acid. So we do have a change in the DNA sequence, which is truly a mutation, but it is silent. We don't see it. We don't see it in the protein. We don't see it anywhere. So we move forward. The next type of mutation, so those were point mutations and types of substitution mutations from those point mutations, is a frame shift mutation. The frame shift mutation causes all amino acids after the mutation to be effective. There are two types, insertion and deletion. And the biggest things that change it are either adding or subtracting two bases or one base. When you add or subtract 3 bases or a multiple of 3, what's going to happen is you're taking out an entire set of codons. Yes, you're changing the reading frame, but you're just taking out 1 or 2 amino acids that you had in that protein. And it will affect that protein, but it's not going to affect it like the full frame shift mutation. So in a frame shift mutation, if we look at the fat cat ate the rat, right? Well, if we add in an A in between the C and the A in cat, we now have the fat ka tat eth ta. Doesn't make any sense. It's not English. Same thing happens in your cells. If we add in a base, it's not being read the same way. And so we have totally different amino acid sequence than we would have had before. So we have a totally different protein than we would have had before. So let's take a look at what's actually happening. Okay, so if this is our DNA base, right? Base is right here. Remember, you're going to need to code for the mRNA base in order to read for the amino acid sequence. The mRNA base is going to be read by your ribosome, which is then going to have your tRNA bring in your amino acid. Now, this codes for histidine. This is the original sequence. If we change one letter right here, and we add in that base right here, everything after that point has changed. We no longer have histidine all the way down, but we start with histidine, 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 and then we have that base added in, and now we have totally different amino acids, which is going to be a different protein. Lastly, we have a deletion mutation. Same thing. We're going to start with that histidine, and then we're going to take out one of these A's. When we take out one of these A's, we're changing the sequence again. And when we change the sequence again, we're changing the amino acid, thus changing the protein. So the big thing that we are doing here is we're changing the protein that we're making. And we're changing the instructions for the protein. And when we do that, and this is when people get disorders, this is when people don't have certain enzymes, this is when people have all kinds of things with proteins in their bodies. So mutations can be good, they can be bad. A lot of people think of them as bad. They can be very, very good. They can help us with evolution. So mutations are not all bad. If you're sitting there thinking about mutations, if you are sitting there with naturally red hair, you have a mutation. Red hair is a mutation. If you are sitting there with curly hair, you have what is called a thymine dimer mutation. So there are all kinds of mutations. They're not necessarily good, they're not necessarily bad. They might be totally neutral, and you can go on living with or without them. So just kind of think about what mutations are, how we get them, and how they're translated into our proteins. Okay, we will talk about this a little more in class.